Here I introduce our research activity on liposomes and how they are used to create artificial cells, which is being carried out at the Nanobio Modeling Lab of the Faculty of Science and Engineering, Chuo University. Now, let's explain the concept of constructing a cell model. First of all, what is a model? It is a partial extraction of the structure and function of the real thing to be tested and utilized. Artificial cells are created by combining molecules such as DNA, proteins, and lipids. You don't have to reproduce everything completely, you just need to construct the parts you need. To create cell models, you first need a container, and liposomes are used for this purpose. Liposomes are lipid bilayer membrane sacs formed by self-assembly of lipid molecules in water. It is the main component of actual cell membranes. Liposomes that are about the same size as cells can be made using various methods. Liposomes in this micrograph was made in our lab. Various molecules can be encapsulated into liposomes. In addition to just encapsulating DNA, proteins, ions, etc., it is also possible to perform their reactions inside liposomes. Indeed, various reactions are taking place inside actual biological cells, so they can be considered as small bioreactors. This photo shows a result of synthesis of a green fluorescent protein in a liposome. Cell models created in this way can also be considered as molecular robots. Cells are molecular machines that sense the environment, make decisions, and react and move. Attempts to combine multiple functions to create molecular robots with more complex structures and functions are becoming very active. Hereafter, I am going to show you our research activities. An important function of cells is their ability to reproduce themselves through cell division. In this movie, multiple liposomes first fuse together using an electric pulse, but then, when left alone, they spontaneously deform to divide. We found that this phenomenon occurs when liposomes are tightly packed with polymers, similar to actual cells. We showed that the membrane deforms inward due to a physical effect called the excluded volume effect, which causes the division-like deformation to occur. In eukaryotic cells, a complex molecular mechanism called mitosis operates to partition duplicated DNA and divide. However, our result shows that membrane division could occur even in a very primitive system. Membrane deformation can also occur in different ways. Actual cells come in many different shapes, and especially the shape of nerve cells is very unique. We discovered that when liposomes are slowly contracted by osmotic pressure, tubes of lipid membrane come out and extend radially, forming a neuron-like shape as a whole. This result also showed that cell morphology could be modeled with a very simple mechanism, and this result was featured on the cover of a journal. Next, we tested what happens when a large molecule, DNA, is placed inside a liposome and the liposome is contracted. DNA shown in green was encapsulated within liposomes, shown in red. As the liposomes were shrunk by osmotic pressure, the DNA attached to the inner side of the membrane and became wrapped, causing budding. This phenomenon was also assumed to occur due to the excluded volume effect. Cells produce many structures in which variety of substances are wrapped in membranes, so this principle is likely to be widely applicable. Finally, we discussed the method for producing uniform artificial cells. Thus far, liposomes have been made by swelling, stirring, or centrifuging lipids in large containers. However, it has been difficult to create liposomes uniform in size. Recently, technology has been advanced that uses microfluidics to generate uniform droplets and uses them to make uniform liposomes. In our lab, we have developed a system that allows droplets to pass through an oil and water interface in a microchannel to produce uniform liposomes with good reproducibility. We would like to utilize this technology for producing uniform liposomes toward the standardization of artificial cell manufacturing. As technology develops, standardization becomes necessary. We will continue to strive to develop versatile artificial cell technology that anyone can use. Thank you for watching the research introduction of the Nanobio Modeling Lab, Faculty of Science and Engineering, Chuo University. If you are interested, please join us in research and enjoy science and engineering together.